So the next step we are going to make is on the ideogram. What is the ideogram? The ideogram is a graphic representation of the chromosome. Okay? A graphic representation of the chromosomes that depicts where the bands are on the chromosomes and where the subbands are on the chromosomes. This is very important because this allows you to know specifically where a particular defect is located on the chromosome, right? And again, you have the short arm, the long arm, okay, short arm P, the long arm Q, right? You have a band, and within a band, you're going to have some sub-bands, right? So you have the band that goes from here till here, but that's not all. We want to know specifically where. So we have to address the sub-band, right? So most areas on the chromosomes are going to be delineated with a pattern of bands, right? So that we can know that, okay, chromosome that we call chromosome number 1 from mom and chromosome number 1 from dad, they have the exact same bands everywhere. They have the exact same subbands everywhere. That's how we know that these chromosomes are homologous. All right. In terms of chromosomes abnormalities, what we call abnormalities, it's basically when there are too many, when there are too little, when there are changes in the arrangement, in the structure of the chromosomes. Now, all these abnormalities are going to lead to, most of the time, to spontaneous miscarriage. I want you to remember that. Spontaneous abortion. I want you to remember that. Okay? So you only have a very limited amount that is viable, right? So that really most individuals are normal. So this speaks to really how our system works well at trimming abnormalities, right? So that these abnormalities are not found persisting in a population. Okay? So nowadays we can see that because of the new technologies, we can now start paying attention to, okay, even when we look normal and when we behave normal, that, well, some problems can happen in the chromosomes without even us knowing it, unless we apply some new technologies that allow us to really tell apart the defects from the normal conditions. Now, what are some abnormalities that we are going to address? We're going to address polyploidy, okay? When we have extra chromosomes for every single of the chromosomes. So pair number 1, we have an extra pair number 2, we have an extra pair number 3, we have an extra until pair number 23rd, right? We're going to take a look at aneuploidy. Aneuploidy, it's basically when you have an abnormal number of chromosomes, in a sense that one of the chromosome is extra or one of the chromosome is missing. If one of the chromosome is extra, in that case, we are going to call the condition a trisomy, right? If one chromosome is missing, in this case, we are going to call this condition a monosomy. I want you to remember that. Then, other abnormalities are deletions, so loss of a segment of the chromosome. Duplication, it's twice a type of particular segment of the chromosome. Translocation, it's basically changing positions, right? From one position to another position. Inversion, it's a form of translocation where you actually have, you know, um, an inversion. So instead of reading A, B, C, D, it's going to read A, B, D, C, right? We are going to take a look at that. The isochromosomes, it's a chromosome with two identical arms, okay? We have ring chromosomes and chromotropsies. Chromotropsies, it's basically one or more chromosomes that shatters, right? All right, so polyploidy, it's not very typical in the human kind, right? Polyploidy is not typical to the human kind. And actually, even more, it's not typical in animals. But polyploidy is common, actually, is more frequent in plants and specifically flowering plants than you do have in other living organisms, right? Some fishes, and particularly in the farming, we can have some salmons that are triploid. We can have some salamanders that are triploid, but this is not very common, okay? And triploidy is going to occur if you have a sperm that has every single chromosome double, or an egg cell that has every single chromosome double, or an egg cell that get fertilized by two sperm cells. While it is very unlikely, nevertheless, accidents happen, right? Okay, so this is an example of triploidy, where you have every single type of chromosome here is represented 3 times. So it's amazing. Well, here it is less than 3. It's the normal amount, but everywhere else it's 3. Okay? So this is a good example of triploidy. Aneuploidy, anu means abnormal, right? So abnormal number of chromosomes, so right here, I'm sorry. Aneuploidy, anu means without, right? Neuploidy, normal, right? Euploid, it's normal, but aneuploid, it's abnormal. All right, so here we are going to have situations of trisomy, extra chromosome, and monosomy, missing a chromosome. And again, most of the time, aneuploidy is going to lead to spontaneous abortion. All right, so aneuploidy can actually cause a mosaic, right? A mosaic because sometimes individuals can have chromosomes that are distinct, right? One is going to be have the allele that is dominant. The other one is going to have the allele that is recessive. So which one is going to be absent is going to actually cause a situation of a mosaic. So aneuploidy occurs because of non-disjunction. These non-disjunction events tend to increase in frequency when a lady reaches the age of 35 and above, right? This tends to increase the possibility of aneuploidy. That's why whenever a lady is 35 and above, they are systematically screened when they are pregnant, right? They are systematically screened for aneuploidy, at least in the United States. All right. So non-disjunction can occur during meiosis 1 or during meiosis 2. When non-disjunction occurs during meiosis 1, all the gametes are abnormal. two gametes are going to have a monosomy condition, while two gametes are going to have a trisomy condition. This is when non-disjunction occurs during meiosis 1. But when it occurs during meiosis 2, two cells are going to be normal. They're going to have the normal number of chromosomes, so they are haploids, while two are going to be abnormal, one being monosomic, the other one trisomic, right? All right. So aneuploidy happens among among humans, right? And aneuploidy typically are going to happen for autosomes chromosomes, right? And autosomes chromosomes are these chromosomes that are not involved in the determination of sex, right? What are some aneuploid conditions that are common in humankind? You have trisomy 21, okay? We have trisomy 18 and trisomy 13, right? And this situations actually leads to syndromes like the Porter syndromes and the Edward syndromes, okay? You can see that these two conditions, extra chromosome 13 and extra chromosome 18, are not very common, okay? Because the individuals are going to die in a relatively early age. So survival rate 1 year after birth is less than 5% for trisomy 13 and less than 5% for trisomy 18, while that survival rate is going to jump to 85% for Down syndrome, right? That's why in the population, you are going to have more people with Down syndrome than any of these other syndromes. All right, now, because of the progress that we've made these days, people with Down syndromes are able to actually thrive, reach adulthood, be able to be inserted in the community and in the society, and doing a specific work and being able to work at different places and holding jobs, right? Here we have an individual with Down syndrome who is painting. Long time ago, they were not able to actually do some specific, hold some specific jobs, and were not taken care of properly because of lack of knowledge and also lack of facilities. Now, there are technologies today when it is possible to turn off some chromosomes. You know that in a female has XX, and we did say in the previous chapters that, well, of the two X's, one becomes a bar body. Knowing that, the question comes to, okay, well, in someone with trisomy, is it possible to turn one of the chromosome 21 into a bar body? Well, guess what? There is a possibility for that now, right? So basically, it is possible to silence the extra genetic information in some individual with trisomy 21. But how does this happen, right? So typically what is going to happen is that in an individual with trisomy 21, we're going to isolate some fibroblasts, and fibroblasts are cells that are found in the loose connective tissues, for instance, underneath of the skin, right? These fibroblasts have extra chromosomes, right? So this fibroblast cells are going to be modified I don't know if you remember the IPS technology, the derived induced a pluripotent stem cell. These cells are modified, right? They are modified to become stem cells, the fibroblasts. They are modified to become stem cells, right? And as they are modified to become stem cells, they are going to be treated with a particular chemical, basically the XIST RNA. The XIST RNA is going to actually go and label one of the extra chromosomes, right? And that chromosome is going to be methylated, right? That chromosome is going to be methylated. And once the chromosome is methylated, then what? Then what? The what? It's that chromosome is actually going to be going to be shut down in terms of gene expression, because methylation causes a shutdown of gene expression, right? So at the end of the day, the heavily methylated chromosome is going to form a bar body, and that chromosome is not going to be expressed anymore. And now, this cell that is modified can now differentiate into forming different kind of cells, and this cells can be actually put in a patient, and the cells are going to thrive and form different kind of cells in the body of that individual. All right. So here we have the case of a baby that was born with trisomy 18, and notice the way, for instance, this baby is closing their hands, right, with the fingers on top of the thumb, right, while the thumb should be on top of this finger here. Okay, so this is typical to trisomy 18. So non-disjunction occurs in meiosis 2 in oocyte, and in that condition, in that situation, the cell is in general not going to survive. Okay, individuals are going to be born, when they do survive, with serious mental disability and physical disability. All right, in trisomy 13, this is going to actually, when the individual is able to develop till birth, right, they are going to have serious intellectual disability. Physical disabilities are going to also be there, right? There are a bunch of deformities that are physical, right, that are typical of trisomy 13. The individual may have some extra fingers, or sometimes extra toes, or sometimes both. All right. Aneuploidy doesn't only happen for autosomes. It also happens for sex chromosomes, right? We can have missing sex chromosomes or extra sex chromosomes. But this is going to basically lead to some conditions, some of them more or less severe, but most of the time they are not going to be severe. All right. So in the normal condition, an individual is going to have an X and a Y for a male that is normal. A female is going to be X and X, so 46XX, that's normal. Now, some females can have non-disjunction, okay? So for non-disjunction, we have XXY. For XXY, this is a male that has an extra X chromosome. XX and an X, we have triple X individuals. We can have sometimes, individuals with X and no Y. This individual with X and no Y are going to have what we call Turner syndrome, while individuals actually that are only Y but no X are not viable. We don't know of any male that doesn't have an X. Okay? So meiosis can occur at different stages of, non-disjunction can occur at different stages of meiosis, right? So now let's jump right here. Some individual can be XXY, just like we saw here. Some individual can be triple X, XYY, only X alone, and XXYY. Okay, so we are going to address different syndromes here. Now, the individual with XO is an individual with Turner syndrome, right? This affects about one individual out of 2,500 females. Now, these individuals are actually going to, most of the time, die, but the one that survive are actually going to exhibit some features, including not a complete development of female features. For instance, their breasts are not going to be very much developed. The menstruations may not occur throughout the lifetime of the individual, right? They're going to have short in stature, right? Webbing at the back of the neck is very characteristic of individual with Turner syndrome. An impairment in hearing can also occur, and these individuals actually are going to have a mosaic, okay, and may have children or may not have children. Most of the time they don't have children. Okay, so the triple X syndrome, these individuals are actually normal, okay? They are normal. They don't have any particular issue. There is no symptoms. They're going to tend to have female exaggerated features, but beside that, really, there is no major issue here. In males with an extra X, these individuals are going to be what we call, they're going to develop what we call the Klinefelter syndrome. In the Klinefelter syndrome, the individual is male, right? They're going to have an incomplete sexual development. In other words, actually, they're going to be males that have underdeveloped male features for an exaggerated female features, right? So that's basically what it means, right? They're going to have rudimentary testes. That's what I mean by underdeveloped male features, right? The prostate is going to be underdeveloped. They tend to have long limbs, large hands, and large feet, right? Some are going to have breasts that are going to be more developed than in an average male. So because of the underdevelopment of testes and prostate, this individual tend to be unable to reproduce, right? We're going to have also individual with double X and double Y. These individuals are going to be associated, actually, the syndrome associated with double X and double Y, it's basically ADD, right? Obsessive compulsive disorders. Some are going to have learning disabilities. Not everyone is going to have learning disabilities, but for sure, the individual is going to not be able to sit still, right? Individuals are going to be infertile. Okay, this condition can be treated with additional testosterone. Okay, the Jacob syndrome. The Jacob syndrome occurs in males where they're going to have exaggerated male features, right? They're going to have exaggerated male features. Great height, for instance, it's one of these features. Persisting acne, even when the individual has been above maturity. So they also can have speech and reading issues. Now, what are some atypical chromosomal structures? The atypical chromosomal structures are going to be, maybe we have too much genetic information on a chromosome, maybe we have too little genetic information on a chromosome. But how does this happen? Well, we are going to see this. Having too many genetic information or too little genetic information can affect the way we behave, the way our health goes, right? The way our metabolism goes, right? Then the mechanism by which this happens is what we are going to address in the next few slides. All right. So, what are these abnormalities? We are going to have that here we have a normal chromosome with the normal sequence of genes. So A, B, C, D, E, F, G, and so on and so forth. We can have deletions. This is when you have loss of genetic information. So for instance, here we have A, B, C. In the normal chromosome, we did have D, E, F, but now you don't have D, E, F. Okay, that's a deletion. Duplication, it's when you have a repeat in the amount of genetic information. So we have A, B, C, D, E, F. And then we have another extra D, E, F, right? So now you have twice the amount of genetic information here. Then we have inverted sequence. Inverted sequence, we technically have A, B, C, D, E, F, but rather than having D, E, F, we have F, E, D. So this segment has been switched from reading this way to reading this way, which is a problem, right? Okay, so deletions and duplication, this deletion can happen de novo, means it happens and never occurred in the parents, right? But in the great parents, but when the gametes are forming, this deletion can occur. Some examples of deletion that can happen de novo is cri du chat. In French, we read it cri du chat. Cri du chat, right? It's basically cat cry, okay? The cri du chat syndrome is typical to the voice, the pitch of the voice in the individual that is reminiscent of a cat that is crying, right? Because of the change in the structure of the larynx, the pitch of the voice changes, right? So this is a deletion that happens. P, remember, P is what? Short arm. Which chromosome? Chromosome number 5, right? So chromosome number 5, short arm, you have a deletion there that is responsible for the syndrome Cre-du-Chat. Okay? You have a duplication. That duplication can occur de novo, right? This is an extra genetic information. We already talked about this. Okay? So we have, for instance, the example of a duplication that occurs in chromosome 15. In that duplication, we see that we are going to have, actually, if we have a duplication here that happens once, okay, we have no symptoms, so no big deal. Then we have a duplication that is going to involve not just gene A, but also gene B. This is type 2 duplication. We have no symptoms. But wait a minute. When we have type 3 duplication, so we have A, B, C, D, E, F, G, H. That is copied twice, so 1, 2. Ooh, what happens is pretty significant. And what happens is that you have poor muscle tone, epicanthal fold in the eyes, right? Small size, intellectual disability, seizures, developmental delays, okay? Curved spine, scoliosis, learning disability, okay? Autism with problem with speech, hand flapping, lack of eye contact, need for routine. So we are literally going from no symptom to all the symptoms, right? Okay, translocation, it's basically when we have a change in the positions, right? And this change in the positions can lead to two major types of translocation that we know of: the Robertsonian translocation and the reciprocal translocation. But what is the difference between these two? In the Robertsonian translocation, well, say here we have Mrs. P, right, with chromosomes 14, chromosomes 21, and Mr. P, okay, with chromosomes 14, 21, and a translocation here between chromosome 14 and chromosome 21, okay? We have a translocation there. Now, when we consider this individual, okay, so Mr. P can produce different types of sperms, actually 6 different types of sperms. This individual, Mr. P, can produce sperms with chromosome 14 and chromosome 21. This is normal, okay? He can produce a sperm with a chromosome with translocation between 14 and 21, okay? So we have a translocation carrier here. We can have a sperm cell that has chromosome 21 and a translocation 1421. This individual is going to have Down syndrome, okay? We can have chromosome 14 and translocation of 1421, okay? A fertilized egg with this sperm cell is going to lead to spontaneous miscarriage. Then we have chromosome 14, okay, sperm cell with chromosome 14, that also leads to spontaneous miscarriage because of the lack of chromosome 21. And here we are going to have a sperm with chromosome 21, and this sperm is missing chromosome 14. That is a problem. We notice that it leads to a spontaneous miscarriage. All right. The reciprocal translocation. So pay attention here. We have homologous chromosomes here, okay? And homologous chromosomes here. So we have these two guys that are going to undergo translocation, and we see a modified chromosome here and a modified chromosome here. And see how there is a translocation here and here, right? Using the fishing technique, we can actually locate some chromosomes that have translocation, right? Okay? How is inversion occurring? The inversion can occur based on actually where the centromeres are located, right? We have the paracentric inversion. In a paracentric inversion, well, notice that we're going to have a recombination that occurs here. And upon that recombination, what do we see? We see that, well, we have a normal chromatid here, an abnormal chromatid here with two centromeres, okay? We have a chromatid here with inversion. We have a chromatid here with an eccentric fragment, right? Eccentric because the fragment is not quite at the center. The center would be here, right? Okay? We have a pericentric. Peri, it's around the center. So the centromeres are not quite at the end of the chromosome. And so where the recombination is going to happen is going to be here, so that you are going to have these different combinations for inversions. I'm not going to bug you with the details here. The isochromosomes, it's basically that, well, you have the two sister chromatids, but how the two sister chromatids are going to split during cell division is going to give you some interesting outcomes, right? Technically, you should have a split right here so that you have one sister chromatid and two sister chromatid, but guess what? It doesn't always happen that way, okay? Doesn't always happen that way. It can actually happen in a way that, boom, you have a separation here so that you have these two short legs that are together. So you have a duplication of the genetic information there. It can happen so that these legs are going to come together. That's what you have here, okay? Or you can be in a normal situation right here, right? So how the chromosomes are separating during cell division is going to play a very important role so that you can have isochromosomes. So different forms of the same chromosome. And then we have ring chromosomes. Ring chromosomes are going to typically happen when you have some deletions on the chromosomes and you have at the end of the chromosomes some sticky ends. These sticky ends are going to come together to actually form a ring, right? That ring, it's actually what you see here. You have a normal linear chromosome and you have a ring here, right? When the ends are chopped up and you have some sticky ends that bring them together, all right? All right, so what are the causes of chromosome abnormalities? Here we are doing a summary. We have, in terms of number, we have polyploidy, you have aneuploidy, right? Polyploidy, when every single set of the chromosome is doubled, okay? Aneuploidy, some of the chromosomes are represented too many or too little, right? Then you're going to have structural abnormalities. You have numerical abnormalities and structural abnormalities. In structural abnormalities, we're going to have deletions and duplications. We're going to have translocations. We're going to have inversions, decentric and acentric chromatids. We're going to have ring chromosomes and chromothropsis, okay? And then we are going to have conditions where individuals are going to receive extra chromosomes from one of the parent. We call this uniparental disomy. Uniparental disomy, it's that, well, you receive twice the amount of genetic information from one of the parents, right? For disomy to occur, you actually going to need that during fertilization, right? Whether the egg did not have one of the chromosomes, but but dad comes with an extra of that chromosome that is missing, right? Or dad did not have one of the chromosomes, but mom has that extra chromosome that is coming, right? So here, non-disjunction can occur, right? In a sperm cell, it can occur in an egg cell, okay? We can have trisomy or loss of the chromosome. In any case, you're receiving double dose from mom or double dose from dad, right? So what are some examples of uniparental disomy? Some examples of uniparental disomy are, for instance, we do see this in cystic fibrosis when, say, dad never had any allele responsible for cystic fibrosis, but mom has the allele for cystic fibrosis, but mom is a carrier. After duplication, right, and non-disjunction, right, well, the affected chromosome is the one that is passed down, right? And at the end, even though mom was not affected, did not have cystic fibrosis, mom was a carrier, but they can actually have an offspring that is expressing the disorder because, well, they received a double dose, right? Okay, so this is a situation that can happen. Another example of that kind of situation is the Prader-Willi syndrome that typically happens when there is a double dose effect that comes from mom, right? And Engelmann syndrome, it's actually the same chromosome, right? Same deletion, but this time it's going to come from mom, right? So the same deletion coming from dad is going to cause the Prader-Willi syndrome. Coming from mom is going to be called the Angelman syndrome, right? So here we have the case of an individual in this pedigree. Mom here is a carrier for cystic fibrosis, so she's not sick, she doesn't have any problem. Dad is completely normal, right? But they have a daughter with cystic fibrosis. How did this happen? Because the chromosome of mom that carried the recessive allele for cystic fibrosis got doubled and ended up in an egg cell that ended up being fertilized so that the daughter has cystic fibrosis. Interesting things happen in genetics, believe it or not. So this is where we are going to stop this conversation. Thank you for your attention. And if you have any question, please let me know and send me your questions. Thank you. Bye now.